Welcome to AP Biology. This video is on Mendelian genetics. Gregor Mendel is considered the father of modern genetics. He created laws of inheritance that form the basis for all we know today in the field of genetics. Before the time of Gregor Mendel, people believed in blending inheritance. They believed that when two individuals mated, all of their genes blended together. So offspring were a mixture, a blend of the two parents. Gregor Mendel proved that this was not true. Gregor Mendel performed studies on pea plants, and he studied 7 different characters. Today we would call these characters genes. Things like height, flower color, pod shape. These are the different characters that he studied. For each character there were alternative traits. For flower color you had purple and white. For height you had tall and short. For pod shape you had smooth or wrinkled. Mendel controlled the mating between these pea plants. He either self-pollinated plants, he crossed a plant with itself, and by doing that he would create true breeding or purebred plants, or he would cross-pollinate. He would mate 2 different types of pea plants together. Here you can see how Mendel controlled pollination. He used a paintbrush to physically transfer the pollen from the stamen of the male to the carpel of the female. And you can see by doing this he could have created true breeding or purebred plants, or he could create hybrids. Hybrids would be mixtures of different plants. Let's look at Mendel's first cross. He began with 2 members of the pea generation. The pea generation is the parental generation, the first generation. And he began with 2 true breeding plants. True breeding purple crossed with a true breeding white. And he created these purebred plants through self-pollination. Based upon blending inheritance, the offspring, which are the F1 generation, should all be a mixture, should be a really light purple color. Well, they weren't. All the offspring were purple. So this immediately disproved the hypothesis of blending inheritance. When you mate 2 true breeding plants together, the offspring are considered hybrids. They're a combination of the 2 plants. In this case, the hybrid retained one of the parental traits. He then took 2 members of this F1 generation, both purple flowers, and crossed them. And what he found in the F2 generation is that 75% of the flowers were purple, but 25% of the flowers were white. So this was really surprising. Not only did the traits not blend, but one trait was hidden in a generation and was able to come out in a later generation. Based upon this cross, Mendel created his laws of inheritance. Mendel said that for each gene there was an alternative version of that gene called an allele. And these alleles account for variations in inherited characters. For instance, here we're looking at 2 chromosomes, and we're looking at the character or the gene for flower color. And you can see that there are 2 different alleles. There's the allele for purple flowers, and there's the allele for white flowers. The specific location of the allele or the gene on the chromosome is called its locus. Mendel's first law is called the law of segregation. And the law of segregation states that the 2 alleles for each character separate or segregate during the formation of gametes. Each egg or each sperm is only going to receive one of the parent's 2 alleles. We know that parents are diploid. Diploid meaning that all their chromosomes exist in pairs. Going back to this picture here, this pair we would consider a homologous pair. So during the formation of gametes, these two alleles would separate, and each gamete, the sperm or the egg, can only receive one of these two alleles. During fertilization, when the sperm fertilizes the egg, one of the mother's alleles matches up with one of the father's alleles. If the organism inherits two of the same allele, we call that individual homozygous. If the individual inherits two different alleles, we would call them heterozygous. This brings us to Mendel's second law. This often is considered part of the law of segregation. It doesn't matter if you consider its own law or not. But the second law is called the law of dominance. And the law of dominance applies to the heterozygote. When you have two different alleles, one of those alleles will be expressed over the other allele. The allele that doesn't get expressed is called the recessive allele. So the heterozygotes only express, they only show the dominant trait. So going back to our cross, the allele for purple flowers was dominant over the allele for white flowers. If you look at this F1 generation here, this is what the F1 generation's chromosomes would have looked like. They inherited one purple allele from one parent, one white allele from the other parent. However, the purple allele was expressed over the white allele. All of these hybrids were heterozygous. They had two different alleles. This will bring up two important key terms. One is genotype and two is phenotype. Your genotype is your genetic makeup. You can have three different genotypes. You could be homozygous dominant if you get two dominant alleles. You could be homozygous recessive if you get two recessive alleles. Heterozygous means that you get two different alleles. You get one dominant and one recessive. Your phenotype is your physical appearance, and you're physically going to either look dominant or you're going to look recessive. According to Mendel's laws, if you're homozygous dominant or you're heterozygous, you will express the dominant phenotype, in this case purple flowers. The only way that you can express the recessive phenotype is for you to be homozygous recessive. So going back to Mendel's cross again, all these white flowers here had to be homozygous recessive. These purple flowers could have either been homozygous dominant or heterozygous. Now, in reality, one allele is not really dominant over the other. An allele can't just suppress another allele. We know that genes code for proteins. Often the dominant allele will code for a functional protein and the recessive allele will code for a non-functional protein. If you are a heterozygote, you make enough functional proteins where the non-functional proteins don't matter. So whether you're homozygous dominant or you're heterozygous, you're going to express that dominant phenotype because you have enough functioning proteins. In the case of the homozygous recessive, you make all non-functional or all recessive proteins, which is what gives you that phenotype. Mendel's law of segregation can be seen in a monohybrid cross. A monohybrid cross is a cross involving one trait. Here you see Mendel's crosses here, and we begin with our P generation. We had a true breeding purple, which would be homozygous dominant. You see here, big P, big P. You have a homozygous white, homozygous recessive, little p, and you can see that all of the members of the F1 generation are big P, little p. They're heterozygous and they're purple due to the law of dominance. In the F1 generation, we then performed a cross between two of the flowers, both flowers being heterozygous. In order to predict the ratio of the offspring, we would create a Punnett square, which is going to be a box. And for a monohybrid cross, the Punnett square is going to contain 4 boxes. Mendel said that during the formation of gametes that the 2 alleles will separate. So if the individual is heterozygous, they could pass on a dominant allele or they could pass on a recessive allele. Those are the 2 combinations that can be found, let's say, in the male sperm. In the female egg, since they're also heterozygous, you can get a dominant P, a big P, or you can get the recessive little p as well. So we've seen the law of segregation take place. We're showing what can be present in the different gametes, right? These might be the sperm, right there. That might be a sperm. This is the other sperm. And then these right here are going to be the eggs. And you're using the Punnett square to figure out all the different possible ways that the father's alleles can match up with the mother's alleles. So one possible combination is that the 2 dominant alleles match up, like you see right here. Here you have a dominant allele match up with a recessive allele. Here, dominant allele with the recessive allele. And this one right here, you would have recessive, recessive. So we could look at phenotypic ratio and we could look at genotypic ratio. Phenotypic ratio would be the ratio of phenotypes. And looking at this cross, 3 out of the 4 offspring, or 75%, would look dominant or be purple, and 25%, or 1 out of 4, would be recessive. They would be white. If you're looking at the genotypic ratio, you're looking at the individual ratios of genotypes. So you would have 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive. So your genotypic ratio would be 1 to 2 to 1, where your phenotypic ratio was 3 to 1. Mendel's third law of inheritance is called the law of independent assortment. The law of independent assortment states that the inheritance of one character does not affect the inheritance of another character. So just because you inherit blonde hair does not mean you're going to inherit blue eyes. Another way to say this is that each pair of alleles is going to segregate into gametes independently. The law of independent assortment can be shown in a dihybrid cross, a cross involving 2 different characters. Here you see an example of a dihybrid cross, and the important thing to remember here is that the inheritance of this Y trait does not affect the inheritance of the R trait. So when setting up a Punnett square to figure out the ratio of offspring, you need to figure out all the different possible combinations of alleles that can be present in the offspring. To do this, we use the process of foiling. So this individual right here is big Y, little y, sorry for my handwriting, big R, little r. So to figure out all the different possible combinations of alleles that could be in the gametes, we FOIL the individual. You begin with first. So you have big Y with big R, and that you see right here. Then you would do outside, big Y with little r, which is what you see right here. Then you do inside, little y with big R. And then finally, last, little y with little r. You want to FOIL the genotypes of both parents to figure out all the different combinations of alleles that could be in the gametes, and then you perform your Punnett square. Usually with a dihybrid cross, they'll only ask you to find the phenotypic ratio. They're not usually going to ask you to find the genotypic ratio. Now, if an individual is homozygous recessive, we know that they're going to express the recessive phenotype. So anybody that has a recessive phenotype, we automatically know that they're homozygous recessive. But somebody who looks dominant, we don't know if they are homozygous dominant or heterozygous. In order to figure this out, we would perform a test cross. In a test cross, you take the unknown individual and you cross them with an individual who is homozygous recessive, who has the recessive phenotype. By looking at the ratio of offspring, you can figure out if that individual was homozygous dominant or was heterozygous. If the individual is homozygous dominant, all the offspring are going to express the dominant phenotype. But if that individual was heterozygous, then only half of the offspring will have the dominant phenotype, the other half will have the recessive phenotype. Another way to figure out the outcome of genetic crosses, we can use the rules of probability. In particular, we use the rule of multiplication. In order to figure out the likelihood of multiple events taking place at the same time, you're going to multiply the likelihood of each individual event. So, if you were to flip a coin, you would have a 50-50 chance of getting heads versus tails. So the likelihood of getting heads is 1/2, the likelihood of getting tails is 1/2. But if you were to flip a coin twice in a row, the first time you flip the coin, you have a 50-50 chance. The second time you flip the coin, you also have a 50-50 chance. But the likelihood of flipping 2 heads in a row, you would take each individual likelihood and multiply them. So you would take 1/2 times 1/2, and the answer would be 1/4, or you would have a 25% chance of flipping 2 heads in a row. This is often used in complicated crosses, such as trihybrid crosses, to figure out the likelihood of offspring. We're now going to move beyond Mendel. All these forms of inheritance that we're going to study, we have discovered after the time of Gregor Mendel. Gregor Mendel's laws are really important because they set the foundation for all that we know today about modern genetics. But when it comes to most human traits are more complex and do not follow simple Mendelian laws. Here we're going to see some other forms of inheritance that Mendel hadn't discovered. All of Mendel's traits that he studied, they all showed complete dominance. With the heterozygote, the one allele was dominant over the other allele. This doesn't always take place. Our first example here is called incomplete dominance. And with incomplete dominance, what's going to happen is the heterozygote is going to be a blend of the two parents. Since one allele is not dominant over the other allele, we don't use a capital and lowercase letter. So in this case here, for flower color, we use the letter C, and here you have a little R above the C for red, and you have the W above the C for white. When you cross a purebred red with a purebred white, all of the offspring are going to be hybrids. They're going to be heterozygous, CR-CW. However, you see here that it doesn't follow the law of dominance. Instead, all the offspring are going to be pink. So they're going to have an intermediate phenotype. If you take 2 pink flowers and you cross them, 2 heterozygotes, you see the ratio of offspring is going to be 1 red, 2 pink, and 1 white. Next, we have codominance and multiple alleles. Both of these forms of inheritance can be seen by studying blood types. When we look at the alleles for blood types, there's actually 3 of them. We have the A allele, we have the B allele, and you have a third allele, which is this lowercase I, which would be the O allele. So when we look at the different blood types, you could be blood type A, blood type B, AB, or O. If you're blood type A, you can be AA, or you could be AO. So in this case, the O allele is recessive to both the A and the B. If you have the B blood type, you can be BB or you can be BO. In either case, you're going to have the B phenotype. So the A and the B alleles are dominant over the O. However, the A and the B alleles are codominant to each other. In codominance, with the heterozygous, in this case AB, both alleles are equally expressed. So for this blood type, you're both blood type AB, right? You express both of those alleles. O is the true recessive phenotype. In order to be blood type O, you have to be OO. When distinguishing the difference between incomplete dominance and codominance, when you look at an animal, for instance, and you're looking at coat color, if you mate a black animal with a white animal and the offspring are gray, we know that that coat color is following incomplete dominance. With codominance, if you were to take a white rat and mate it with a black rat, all of the offspring would be white and black. Both alleles are being equally expressed. Pleiotropy is when one gene has multiple phenotypic effects. So in many cases, one gene does not just perform one function in the body. It does not just control one thing. It can control many things. A good example of pleiotropy is when you're looking at genetic diseases caused by a faulty gene. There's never just one symptom. There's always multiple symptoms because that one gene is performing many functions within the body. Another complex form of inheritance is called epistasis. Epistasis is when a gene at one location affects the expression of a gene at a second location. The best example of epistasis is with albinism. Here you see in this example that we have two different genes. We have the gene for mouse coat color, and the two different colors you could have are gray or tan, and you have a second gene for pigmentation, the color gene, which is C. Now you can see here that gray is dominant over tan. However, if the individual is homozygous recessive for the color gene, it doesn't matter what the other gene says. It doesn't matter if it says it should be gray or it should be tan, that mouse is going to be albino. So the expression of the color gene is directly affecting the expression of the other gene for the coat color. Many genes do not exist in an either-or basis. Many genes exist, or many traits exist along a continuum. You don't just have tall and short. You don't just have brown or blonde hair. You don't just have brown or blue eyes. For many characters, the traits are found in a continuum of phenotypes. We call these characters quantitative because they have the additive effect of many genes. So in this case, you don't have just one gene controlling a trait. You have many genes, 3, 4, 5, 6, or many more. We call this form of inheritance polygenic inheritance. Polygenic inheritance is going to be responsible for any trait that we know of that varies along a continuum. Some of these common ones are going to be skin pigmentation, height, eye color, hair color. These are all polygenic traits. Many genes all interact with one another to form your phenotype. It is extremely difficult to predict the inheritance of any character that is polygenic. For most genotypes, there is not a rigidly defined phenotype, but instead there's this norm of reaction. There is this gradation, there's this range of phenotypes that fit with a very specific genotype. The reason for this is that the environment plays a role in the expression of your genes. In some cases, there is no norm of reaction. For example, blood type. Whatever your genotype is, that's what your phenotype is. If your genotype says AB, then your blood type is AB. But for other traits, the environment plays a large role in the expression of that trait. These traits in which the environment plays a large role are called multifactorial characters. Here we see an example on the bottom right. Both of these flowers are hydrangeas. Both of these flowers have the exact same genotype. However, they have a different phenotype. That's because the color of the flower is determined by the pH of the soil that they live in. So you see that the environment plays a large role in the expression of this gene. So for this plant here, the norm of reaction for this genotype is very broad. It's very wide, and the environment plays a large role in it. In order to follow the inheritance of a trait throughout a family, we use an analysis called pedigree analysis. Pedigree analysis is basically a family tree that shows the inheritance of particular traits. We can use this to determine the likelihood of somebody inheriting a trait, and we could use this to figure out if somebody is homozygous dominant or heterozygous for a trait. With a pedigree, squares are males, circles are females. Typically in a pedigree, if the circle or square is filled in, that means that that individual has that trait. If the circle or square is open, that usually means that that person does not have that trait. However, you should always look at the directions before answering a pedigree problem. You can see here that these two individuals mated, and these are all their offspring. This individual here, this girl here, married this guy, and these are their offspring. The last thing that we're going to look at are genetically inherited disorders and methods used to test for these disorders. The first group of disorders are recessively inherited. If you're homozygous dominant or you're heterozygous, you do not have the disorder. However, heterozygotes are called carriers because even though they don't have the disease or the disorder, they are carrying the recessive allele and can pass that recessive allele on to their offspring. The first recessively inherited disorder is cystic fibrosis. With this disorder, the dominant allele codes for a functional chloride channel protein. The recessive allele codes for a non-functional chloride channel protein. If the person is homozygous dominant or heterozygous, they have enough functional chloride channels where the individual is normal. They don't have cystic fibrosis. If the person is homozygous recessive, they have all non-functional chloride channels. As a result, chloride ions cannot enter or leave the cells. Mucus is going to build up in the respiratory tract and the digestive tract, and the person usually lives to about the age of 30. With sickle cell anemia, the gene codes for the hemoglobin protein. Hemoglobin is a protein found on the red blood cell. The dominant allele codes for a functional hemoglobin protein. The recessive allele codes for a non-functional misshaped hemoglobin protein. With sickle cell anemia, if the person is homozygous recessive, they create red blood cells that have this sickle shape. As a result, their red blood cells cannot carry oxygen very effectively, and they have a range of symptoms, a range of problems. Another recessively inherited disorder is called Tay-Sachs disease. Tay-Sachs disease, the gene codes for an enzyme in the brain that digests lipids. The dominant allele codes for a functional enzyme. The recessive allele codes for a non-functional enzyme. If the individual is homozygous recessive, they have all non-functioning enzymes. They can't break down lipids in their brain, and as a result only live to about the age of 3 maximum. Now, Tay-Sachs is recessive at the organismal level. It's actually incomplete dominance at the molecular level. If the individual is a heterozygote, they're actually making half functional proteins and half non-functional proteins. So their brain is only able to digest half the amount of lipids. So there you see where it's incomplete dominance. However, digesting half the amount of lipids is still enough to allow that person to live. Going back to sickle cell anemia, at the organismal level, sickle cell anemia is recessively inherited. But at the molecular level, it's actually codominant. If the person is heterozygous, they're actually going to make half sickle-celled red blood cells and half normal red blood cells. However, they have enough normal red blood cells where they don't have sickle cell anemia. They have something that we call sickle cell trait. And the reason why we say that they have sickle cell trait is that they do suffer from some symptoms at areas like high altitude where oxygen levels are lower. With any recessively inherited disorder, mating of close relatives, inbreeding, tends to bring out these harmful recessive traits. Not all disorders are recessively inherited. Some disorders are dominantly inherited. One example is achondroplasia, dwarfism. That means that all normal-sized people are homozygous recessive. Someone with dwarfism could be either homozygous dominant or heterozygous. Two people with dwarfism can produce a normal-sized child because they could both be heterozygous. Another example of a dominantly inherited disorder is Huntington's disease. This is a disease of the nervous system. The sad thing about Huntington's disease is a person doesn't know they have it until about the age of 30. So usually they pass on the lethal dominant allele before they even know that they have it. With Huntington's disease, the person goes through an extreme neurological degradation. Their brain breaks down, and as a result, they suffer from many psychiatric problems. There are several tests out there that we can use to identify these disorders or predict the likelihood of passing on these disorders to your offspring. One thing we could use is just the probability rules, right? If two individuals are carriers and it's a recessively inherited disorder, we know that they have a 25% chance of passing on that disorder. We could also use a pedigree analysis. A pedigree analysis can also be used to identify the genotypes of individuals in a family and predict the likelihood of passing that lethal gene on. There are several genetic tests available today used to identify carriers. We have genetic tests for over 2,000 different genes. We will learn about this later on. We use a process called gel electrophoresis in order to test the genes to identify individuals who are heterozygotes. There are several forms of fetal testing that are available, testing the fetus in the womb. Here you see the two different types. One's called amniocentesis and the other one's called chorionic villus sampling. Both of these procedures could be invasive and could potentially harm the baby. They will be performed if there is a reason to perform them, if the doctor thinks that there's a possibility that this offspring could have a potential problem. Out of the two, amniocentesis is less invasive. What happens is the doctor takes a sample of the amniotic fluid surrounding the baby. From that amniotic fluid, there are fetal cells, and the doctor can do two things. One is that they can perform genetic tests on those cells to identify any genetic disorders, and the other thing that they could do using the cell is form a karyotype. And they're going to perform a karyotype to look for disorders like Down syndrome. Chorionic villus sampling is much more invasive than amniocentesis, but can be done much earlier in pregnancy. During CVS, the doctor actually takes a sample of the placenta, and from the placenta, the doctor can obtain fetal cells. Again, using those fetal cells, the doctor can perform genetic tests and also can perform a karyotype. Again, both of these procedures are potentially invasive. They would only be done if the doctor thinks there might be a problem. Amniocentesis is the less invasive of the two, but has to be done later on in pregnancy. Chorionic villus sampling is more invasive, but can be done earlier in the pregnancy, between about the 7th and 8th week of pregnancy. Finally, when a baby is born, doctors perform a newborn screen. They are looking for one disorder in particular called PKU. PKU stands for phenylketonuria, and it is a cause for extreme mental disabilities. An individual that has PKU cannot break down phenylalanine. However, today this is not really a problem. If they detect PKU, they put the baby on a diet that is low in phenylalanine, and it corrects the problem. This concludes our video on Mendelian genetics.